Great, thank you so much. It's such a privilege to be here. So this is mostly what I'd like to discuss today is work that I did at WashU in St. Louis as a graduate student in Jeffrey Gordon's lab, but also to touch on other work in the literature that's done by colleagues both in the lab and elsewhere, looking specifically at nutrition and undernutrition as it relates to host metabolic physiology and growth. This is an enormous topic and one that I don't think we can cover well across the entire spectrum of microbiota disease associations, but we'll focus on nutrition. And I'm currently based at the Massachusetts General Hospital. And again, thanks for the invitation here. So nutrition is really one of the major goals of the century. And it's, when we think of inequalities, it's quite unevenly distributed across the globe. So when you look at, there are about 34 countries that harbor the majority of the burden of undernutrition. And it contributes to a significant proportion of under 5 mortality. And you know, one thing that's really important for us to understand as I go through this talk is how do we define nutrition? And the way that the World Health Organization helped us frame this concept is by measuring height and weight across a very large number of children. And it's a relatively simple concept. They picked 5 countries, actually 6 of them, from many different cultures, mostly like income that was above the poverty line, in about 8,500 children. And they came up with what is a single standard that everyone uses in every clinic, particularly in the developing world, to provide a good description of where a child stands in. And what this does is it gives us some criteria to say that, hey, this kid is actually severely malnourished. Other than the obvious picture here where you can see that a child is emaciated, there are particular metrics, including the weight for length Z-score, less than 3 standard deviations below the median. That's severe malnutrition. There are other clinical characteristics, including bipedal edema and a low mid-upper arm circumference. And for background, we treat the malnutrition with food, which is probably obvious, but the types of foods that we use are energy calorically dense. They include peanut-based foods, as well as, in some parts of the world, including Bangladesh, where I'll discuss, local ingredients that are also calorically dense. How these influence the microbiome is unknown. So the major hypothesis going into all of this is that undernutrition isn't caused entirely by food insecurity. And we know this from twin studies where their discordance shows that this can't be explained entirely by, when we compare dizygotic to monozygotic twins, it's not entirely explained by genetics, by nutrition. We think that the gut microbiota plays a central role here in that it feeds into multiple different areas that could influence malnutrition. So we know that food insecurity plays a role and that genotype could also play a role. But infection with enteropathogens, diarrheal disease is a major contributor. Absorption, including conditions like environmental enteropathy, and of course, immune susceptibility to infections plays another role. So we think that the gut microbiota plays a multi-effect role that ultimately worsens malnutrition progression. I don't really have to introduce this after the lovely talks we've had today, but the one thing I want to emphasize is that we think that food shapes the balance of power in this community, as well as other factors. Food, including breast milk and other considerable functional foods. These can influence absorption of nutrients, metabolism, immunity, and vaccination. And the most central part, and if there's one thing that I'd like you to take away from this talk, is that if we start thinking of these human gut microbes or the gut microbiota just as another organ, this is just like your liver, just like your heart. It's developing and it has certain functions that contribute to host development. We know this from very careful studies conducted in mice where you can raise animals germ-free and colonized with specific populations, that there are very specific elements of our biology that can be influenced by bacteria. Here I'm showing just as an example of angiogenesis in the gut. And this is a carefully constructed study where germ-free mice colonized with, even after 10 days, either with a complex mixture of microbes or a single bacterium, stimulate blood villus network formation. And some of this is dependent on innate immune cells, including Paneth cells. I can't list a whole array of different functions, but certainly gut barrier function is influenced by the microbiota. We know that regulatory T cells and other immune cell populations are also modulated. Intestinal motility, and we've already talked extensively today about short-chain fatty acids, TMAO, tryptophan, and bile salt metabolism, just to name a few, including modulation of neurologic status in specific conditions. The one factor beyond diet that probably trumps diet, in my opinion, is age. It's obvious, but it's worth mentioning that age is the major factor shaping the gut microbiota during early life. And we know that from very early studies looking at unsupervised measures, where each point here represents a sample collected from a child, either in Malawi, Venezuela, or the USA. And you can see that in the period of 2 to 3 years, where this y-axis represents the distance to adults, you get closer to adulthood in a 2 to 3 year frame. Keeping in mind that this study is single snapshots of time of different individuals, so no individual is sampled more than once in this study. Which begs the question of how do individuals develop through time? And that was raised in our last talk by Megan, is the individuality of the microbiome. So to study this, we collaborated with colleagues at ICDDRB. So this is an exemplary organization, particularly in the public health field, where it was here that oral rehydration therapy for cholera was discovered and now is implemented worldwide as life-saving therapy. Mirpur is an interesting place because it's an urban slum area. It's extremely densely populated. So the population density is in the 70,000s, so that's about 4 times New York City, to give you an example. And incredibly, despite that density, the population is incredibly well followed by this organization, ICDDRB. Their mothers, children, and families are able to be followed on a long, like years long basis. So we partnered with ICDDRB in cohort studies where children, we'd receive fecal samples in collaboration with them and perform sequencing and do some of the analyses that have been described to characterize the microbiota composition in fecal samples. But most importantly, we wanted to ask a very simple question, which was, if a healthy child shows up to the clinic, or can I take that sample and accurately predict the age, knowing nothing else about that child, what the age of the child would be? And can we use that as a way to create a biomarker or a computational biomarker of that child's gut microbiota development? So we used a random forest method, essentially a decision tree based method, to identify specific bacterial strains at the OTU level that can serve as markers of the aging process. Keep in mind that this has non-parametric assumptions and is advantageous for the number of data points that we have relative to predictors. And the disadvantage of this is that it's a little bit of a black box nature algorithm. The other really important thing about this is, before we got to this point, there was this hype in the microbiome field that with the computational deepness of our sequencing and the computational sophistication, that there's so much variation between each child from time point to time point and between children that we won't be able to make sense out of temporal progression. So we took these healthy infants and we applied this method to just perform a simple regression where y is the age of the child in a known training set, and we took the relative abundances of each type of OTU that we sequenced. And the result of that is we created a sparse model, a signature per se, of where over here, this is a heat map of the abundances of different bacteria, and we can create a signature of what looks like a temporal progression. And the important thing is that you can prioritize the number of taxa in terms of how important they are for the model. So at the top of this list is Faecalibacterium prausnitzii, which was discussed earlier today. And what that means is that if we were to randomly manipulate the values of that bacterial taxon, it would have a major deterioration on the overall accuracy of the model. And finally, we can perform some cross-validation and get to a minimal set of taxa that allow us to perform a reasonable model. So just to summarize here, in a training set on the far left, we have that pattern through time, and each point over there represents a fecal sample. And then we can go to a completely different set of children and ask, can we now apply this model and how does it function? And there was a reasonable fit to samples that are obtained from completely unrelated children in Mirpur, as well as healthy twins and triplets, a separate study that we did. And just to summarize, this is model training and separately applied to other children. We've also applied this, interestingly, to children raised in other countries under different conditions, including Malawi and Peru. And we found, surprisingly, a decent application of the model. Then, in a way that was almost inspired by the World Health Organization, we asked, can we then take this measure and derive certain metrics? So some of the metrics that we defined is to take the microbiota age of a given child, in pink at the bottom over there, and ask where does that measurement fall compared to a cohort of healthy peers, health defined here by those anthropometric measures. And then in a similar way, can we derive a z-score-based measure on a month-by-month bin? So once we derived these measures, we wanted to ask, what are the environmental factors that affect these metrics? And Megan pointed out from more recent studies that in other populations, there's been extensive associations with feeding, birth status, and so on. We interrogated the same parameters here, but we didn't have, most of the children here were from vaginal deliveries. But what we did find is that the diarrheal disease was associated with a significant, at least statistically, drop in the maturation of the microbiota, and that recovered over time. It's important to note that in this population, the burden of diarrheal disease is about 5 diarrheal episodes per year. And we didn't find a specific association with any particular type of enteropathogen that we tested.
Can you go back?
Yeah.
Is that showing that they actually overcome? 4 to 6 months?
It's, I wouldn't say they overcome. It's not statistically significant in terms of comparing to 0. But this trend, I would call it more of a trend than they've become higher. It does trend that way. But this is a relatively small study as well. It's about 25 children, 12 sets of twins, and 1 set of triplets. So
is that adjusting for the age, for example?
Yeah, so this is all adjusted for age, basically, and age is included in the model. Yeah. So the important thing, though, is that we also tested for formula in the diet, and that did have a small positive effect, but statistically significant, but didn't seem that large.
So this is basically like catch-up growth. But then if anything, you're kind of overshooting.
Yeah, I don't know if I would overinterpret that last point. It's hard to really say. Yeah. Yeah, exactly. Yeah, and it might be in that setting of the perturbation. So, having studied that structure in a relatively healthy population of twins and triplets, we wanted to study children with severe acute malnutrition. So I already defined this for you, but when these children are treated, they show up to the hospital and they're stabilized with the milk-based diet, oral rehydration, parenteral antibiotics, and administration of the therapeutic foods I also mentioned to you. And those are the foods, peanut-based and khichdi and halwa. So we applied this, and this is sort of the environment that these children are in the hospital. It's close quarters between different children who are admitted. The mothers play a very strong role in terms of they're right there. There's a lot of communication between the two, and diarrheal disease burden is extremely high in this setting. So the study that we applied these metrics to was a randomized trial comparing the peanut-based diet in orange to the local khichdi and halwa diet. So that's akin to foods that you may see in a South Asian restaurant, but more high dense, more sugar. When they compared the clinical parameters of these two treatments, they found that in the acute setting, so on the left is the peanut-based arm and on the right is the leafy green local-based diet, they saw an improvement in weight for height at the end of the intervention and persisting for several months. The issue being that these children leave the hospital and they never really come back to a level that's similar to the national average. So then, with the more I would say the more worrying thing about the fact that these interventions had no effect whatsoever on height for age, so severe stunting. And stunting is one of those conditions that's actually associated with poorer long-term development in terms of cognitive ability, lifetime income. There's several associations with stunting that are of a poor prognosis longer term. When we applied the same metric to children, where we compare healthy children to the two arms, we find that at admission, children have a severely immature microbiota compared to healthy controls, and that even following the dietary intervention in each arm, there is a significant gap between where healthy children lie. In the months following, we see that they don't quite return to normal, although there's a trend in the peanut butter arm as well as the khichdi halwa arm is more flat. The follow-up period of time was about 6 months in this study, and you can actually see that quite visibly on the curve as well, each of those sample being a fecal sample collected in the follow-up period, that there's a big gap in the microbiota, and there are hundreds of taxa that we identified that are deficient in these children. The other more obvious question is that given that they were receiving antibiotics during this period of time, in a subset of children where we were able to sample the children before they received antibiotics, they also had an immature microbiota at that time point. Interestingly, that microbiota was dominated predominantly by streptococcal species, and we found that antibiotics stimulated Enterobacteriaceae, but that also recovered following the antibiotic and dietary interventions. So the conclusions here are that these two different foods, they produce an improvement, but it's not sustained in their microbiota, and there's significant gaps in their microbiota. They also improve weight, but they don't improve stunting and possibly other longer-term outcomes. So it raises the possibility that more prolonged food-based interventions of varying composition and addition of gut microbes may be beneficial. And the predictors, including the Faecalibacterium and some of the other species, may themselves serve as therapeutic agents in these contexts. And then this is more of a pictorial summary, but I think it raises important questions about where those bacteria come from. So the maternal input that we have discussed, how food patterns might influence those different bacteria, and what the impact of these changes are on brain, bone, immune system development. The other question that comes to mind is, are these changes causal, or casual. So there have been studies by colleagues in the lab, including Michelle Smith and Tanya Yatsunenko, who looked at Malawian twin pairs who are discordant for kwashiorkor. And the kwashiorkor is a variant of severe acute malnutrition that's more common in Malawi. And what they see here when they transplant the microbiota of each co-twin to a mouse that is fed a Malawian diet, they see a discordance in the weight even after 2.5 weeks. That's rescued by the peanut-based diet, but not durable. And it's important to know that even over a longer period of time, the Malawian diet doesn't seem to support weight growth in these animals. You see a similar pattern in the human data, where they see that in the healthy co-twins, there's a maturation that is not seen in the kwashiorkor co-twins. This is true also of children who aren't fortunate to have a twin. In singletons with chronic forms of undernutrition, Laura Blanton in the lab transplanted several donors from Malawi and saw a pattern of decreased both weight and lean mass in the stunted and underweight donors. So there is some component of transmissibility to these phenotypes. Another study showed, when we looked at maternal milk composition in these children, the same population, it was found that the human milk oligosaccharide composition differed in a particular sialic acid dominant milk oligosaccharide. So a hypothesis was constructed where sialylated bovine milk oligosaccharides were found to have a growth benefit on top of a Malawi diet. So that's the top slide over there. And interestingly, this is entirely dependent on the microbiome, because in germ-free animals, providing sialylated BMOs doesn't provide any benefit. And fascinatingly, with bone micro-CT, we can actually, Mark Charbonneau, who did all this work in the lab, showed exquisitely that there's differences in trabecular bone as a result of these phenotypes. So that last example is intriguing, and it speaks towards how interventions of microbes in collaboration with a fructooligosacchαρide can have a huge impact in our mouse models. For the most part, probiotics, I know, which we'll be discussing, have been relatively disappointing. But a recent study showed that symbiotic in the form of Lactobacillus plantarum and a fructooligosacchαρide could have a major impact on sepsis in a neonatal population in East India. So this is almost a 50% reduction in sepsis from an early intervention to a naive immune system. So very interesting possibility. And then finally, where does this take the field forward? We talked about that intervention, but do we need to go back in time and rediscover what aspects of a diet, particularly a traditional diet that's been passed over generations, can stimulate microbes of a particular community? So there are certainly studies that are ongoing now to disentangle local foods and local ingredients, including chickpeas, peanuts, and ask how do these specific ingredients affect those specific bacterial species, and how can these combinations be used to repair the microbiome. And I think a lot of this goes back to how do we think about our future and our future children and their interactions with microbes. We talked about heritability. And how do we change our agriculture productions and antibiotic use to create a more sustainable future? Mirpur, Dhaka is a good example of where population density and resource limitations is similar to where the whole world will be 10, 20 years from now. So our studies there, I think, are quite informative. I'd like to just finish on 2 points. One is to acknowledge the Gordon Lab and ICDDRB, in particular at ICDRB, Tamid Ahmed, who's a leader, a nutrition expert, Rashidul Haq and Saida Haq, who are also extremely primary in a lot of the work that I've presented today. And to touch a point here on inequality, I've already addressed sort of at Mirpur how this was a scenario where we could study the microbiome in an environment where resources are quite limited. It forces me to consider also the inequality of microbiome science as we know it. I think that one thing that's been a real privilege is to interact with colleagues from around the world, and it's amazing to them as well how much they're interested in learning about the microbiome and have the capacity to both sequence and cultivate their own microbiomes. It begs an interesting question as to who owns these microbes, because these microbes are owned by all the different communities that we sample. And when we think about funding agencies, research bodies, and collaborations, I think it's incredibly important to consider the ownership of these microbes and to have conversations with including anthropologists as well as ethical bodies to understand how these research studies will translate to clinical applications. I'm ready to go further with questions, but I will like to point out that one really encouraging thing about this last study is that the patents related to that intervention have been licensed back to the community, and so all future findings from this will benefit that community in East India. So I think it's a good example for us as scientists going forward. Yeah.
Good. Questions?
So really, really interesting work. I was wondering if you looked at modeling the maturity index on metagenomics, if we could try to find out, there's this idea that microbes are important for early immune development, so can we find transporters or immune interacting molecules or metabolic factors that could give us some sort of insight into that?
I think that's a fascinating and brilliant idea. There's no reason why you couldn't apply this method to almost any time series based analysis of the microbiome. The only challenges are, at least at the time of the study, are the economic considerations. 16S is the poor man's way of assessing the microbiome at this point, and you could certainly apply pie crust and other methods to guesstimate at what functions are there. However, to metagenome sequence the time resolution that we did across that many children would be financially extremely prohibitive, but as sequencing costs get cheaper, I think that's a wonderful idea. The other approach that we are thinking of taking or took later on was to try to cultivate or isolate the microbes that we are seeing change through time and sequence their genomes in a very methodical, sort of thoughtful way as to what are the genes in the Faecalibacterium in Bangladesh and how do they compare to those in Malawi, and look at redundant functions across those isolated species.
That finding of the maturational age change is really striking to me.
Yeah.
And I'm wondering what we know about that. Like, is that primarily driven by the host? Is that driven by the ecology and the interactions between the microbes? And I'm partly wondering because of that phenomenon of catch-up growth, or catch-up maturation, right, that you showed.
Yeah.
A lot of things do that. Like, so body growth does that. When you go through a period of nutritional stress, you stop growing, you experience catch-up growth.
Right.
And that's because there's some kind of a target-seeking process that's internal to the body. What underlies that similar kind of target-seeking in this ecology of microbes?
I think amazing observation. It's the question of resilience in the system following that disruption. And I showed two different examples. One was the diarrhea, where we believe there is resilience in the system. There is a capacity for the microbiome to return to what would be an age-appropriate configuration. But there are scenarios where you're beyond that point of return, at least for the time period that we observed. The fact that we're seeing children across different geographies, culinary histories, traditions, breast milk composition that have a similar progression, to me favors a host process. And that's why the genetic studies are so fascinating that we talked about, what drives Faecalibacterium in the gut, what are the factors that make it have to return to its abundance level. So host factors are definitely immune factors as well in terms of controlling, preventing overgrowth of, say, pathologic organisms is another possibility.
Can I have a quick follow-up?
Yeah.
So do we have any sense for functional implications of that? So let's say the children can be delayed
Yeah.
because of that diarrheal episode.
Right.
And then let's say the diarrhea is gone, and have a period of catch-up.
Yeah.
Is there a functional consequence of that? For being like a
Right.
4-year-old that has a 3.5-year-old?
Yeah. So we don't know for sure at this point. We've looked at oral polio vaccine responses and some other surrogates for immune dysfunction, but I don't think we have a slam dunk that this in humans is, other than the mouse studies where we can extrapolate from those phenotypes, we don't know in humans what the consequences are of being a 5-year-old walking around with a deficient microbiota. And to be honest, those children, we haven't even had those studies to follow them to know. I would be very interested to know, are their rates of diarrheal disease the same? Are they responding to vaccine? How are they doing in school? There are all kinds of interesting measures over there. And then if you're in the world of probiotics, transplantation, does that affect those outcomes?
If I could add on to that too, I think what we consider normal is certainly a moving target from birth to adolescence and adulthood, and there are data out there showing that fecal microbes taken from, say, 6-month-old infants, under the right conditions, can have a greater growth capacity when transplanted into a mouse versus fecal microbes taken from, say, an 18-month-old. And the microbial functions that have been detected in some of these age-related microbiome patterns, younger children seem to have more microbes that may have capacity for vitamin biosynthesis, for example, versus adults and elderly people. They don't necessarily have the microbial functions that are theoretically associated with nutrient harvest, vitamin biosynthesis, and they may have more microbial functions that could be associated with inflammation. It's a lot of hand-waving because metagenomics is still fairly new in the grand scheme of this, but I think it's not as crystal clear, black and white. Just as many people believe that a highly diverse fecal microbiome is a good thing. It's not a good thing in all instances, as we saw from exclusively breastfed infants. But there are certain microbiome functions that are associated with younger children that may be beneficial in certain areas. So I don't think we should just reflexively want to push them towards microbiome development, without really understanding what it is they might be leaving behind.
Right. And just to add to that, I think, can you develop too fast in an inappropriate setting? Is sort of like is there an appropriate cycle, or if you miss that critical window, does your organ development also suffer? Yeah.
Yeah, as a follow-up on this, so in your nutritional interventions, you didn't help much with the stunting.
Yeah.
And so you still see lag behind the maturity of the microbiome.
Right.
So the intervention didn't actually help with the maturation of the microbiome.
Right.
So if you do microbiota transplantation, for example,
Right.
would you combine this with the nutrition intervention? Would you have
That would be our hope. The only example we have of bone growth per se was in the context of that SBMO intervention, but we haven't really found a microbiota combination that that either has a discordant, like from a transplanted standpoint, has a discordant bone growth. So there are two possibilities. One is that stunting is from an earlier insult that's very hard to recover from. And the other is that we need to find a combination of microbes and diet that would promote bone turnover and growth. So I think that is the most biggest challenge in nutrition. That's probably on the minds of everyone at every funding agency in nutrition, is how do we fix stunting? And it's the only measure that's been associated with poor cognition and long-term development. So it's not easy, and I wouldn't attribute it entirely to the findings of the microbiome maturity and the stunting, but more work is needed.
That's very interesting. I heard that so for the bone development,
Yeah.
Some children they actually become short and they remain short. They cannot grow
Right.
because there seems to be some how to say that, too early maturation of the bone. So the bone stop growing maybe under very severe malnutrition conditions.
Right.
So they become mature. The bone becomes mature, and then they no longer can grow.
Right, and that's been the sort of thought in the field for decades, actually. The only person who thinks that isn't the case is Andrew Prentice, who's done some work in the Gambia, where he's followed children for up to like the age of 10 years. And what's interesting is that he has some hope that even after the 2-year point, it's possible for children to catch up. So there's still some plus or minus in that discussion. But I would speculate that you're absolutely right in that the earlier you address stunting, the more likely you will be to alleviate that.
Oh, really nice work and nice talk. I'm interested, so in the microbiome age, and sorry if I missed it, is it adjusted for breastfeeding, or are the breastfeeding patterns different enough, or are the patterns pretty similar? So how do you know that it's the age and not the weaning, for example, that's driving the shifts?
Right. So breastfeeding in Bangladesh, most of these children are breastfed at least up to 12 months, actually, in this population. Granted, there's a lot of variation within the quantity of breastfeeding, the quality or the composition of breast milk. In the studies of the MAZ, we don't account for breastfeeding at all in making that calculation. It's just age adjusted. And our training set population has an average time of weaning that's pretty comparable to the rest of the population. So what we don't see is, as you had shown, and granted this is a smaller study in a different population, we don't see a clear, like this is breastfeeding and this is weaning, like to solid foods. The other thing, I don't have children, but anyone who does says that, going to table foods isn't just like the child wakes up one day and they're like, I'm going to have family food on the table. It's a gradual process. And so from a metadata standpoint, it's really hard from this small data set to say that breastfeeding definitely had an effect. At least statistically, we didn't see that.
Are the patterns similar? It sounds like you've done work in Malawi and Bangladesh. Like, are they similar in those two different contexts?
So in Malawi and Bangladesh, we didn't see strong patterns of diet or breastfeeding, and that's been the challenge as we go into these data sets. Even in twins, we haven't seen a strong relationship between that. And maybe in part due to the quality of our metadata and the patterns of the practice in these locations. But we haven't seen a strong connection between breastfeeding status, at least plus minus, or specific types of foods that they are being weaned to. The only hit we had was a diarrhea. Yeah.
So there's this idea that the microbiome early on in life is priming your immune system, right? And so your early exposures really affect how your immune system is going to behave later.
Right.
And I'm curious if you, or if anybody else has used this model to look at in healthy people, but are children who have slightly younger microbiomes, are they more then prone later in life to autoimmune disorders, Crohn's disease, things like this?
Right.
Like, if you're not challenging your immune system well enough early on, are you able to then see these effects later?
Yeah. I'm not aware of any literature doing specifically with autoimmune conditions. The only studies that I've seen that have used this are in the setting of obesity. There's been some suggestions in a Singaporean cohort that early hypermaturation, if you'd like, is associated with obesity. In that population, they used fat pad measurements to look at that, and nothing in the sense of autoimmune. But it would be interesting to look at all types of profiling, including cytokines and other parameters. Yeah. Great.
And I have a comment about this inequality, disparity on microbiome research in the developing world. And in one of the symposium I went to, I met a colleague from Pakistan, for example.
Yeah.
And they are so much in need of the microbiome tools and expertise and to look at their populations. But it's simply not there. So that's actually would prevent them from getting access to all these kind of new knowledge and new potential solutions. But also this metadynamic sequencing, the sequencing part per se is cost is decreasing very rapidly.
Right.
But the cost for analyzing the data is skyrocketing.
Yeah.
So we should consider both.
Right.
So it's not just reading the sequence part.
Of course.
Yeah.
Yeah. And also the opportunity, if we address these inequalities, for more real-time solutions. If I was a pediatrician in the ward with a malnourished child, can I use these measures to decide, should this child go home now, or should I wait like 3 more days, or give them some probiotics? And would that ultimately lead to better clinical care? And
we need a very highly time-sensitive technology,
Yeah.
Almost online analysis.
Right.
Yes. Thank you so much.
Thank you so much.
Thank you.
Thanks.